Hi friends. Welcome to a new learning video on PharmaGuideline. In this video, we will discuss the procedure for disinfectant validation. Disinfectant validation is the process of determining whether a disinfectant is effective in cleaning a surface. The purpose of this process is to ensure that the disinfectant is effective and does not cause any harm to the environment or the people using it. There are three main types of validation: efficacy, toxicity, and residual effectiveness. These tests help ensure that a disinfectant is effective at cleansing and sanitizing areas, minimizing the risk of exposure to harmful bacteria and fungus. Efficacy tests measure how well the disinfectant cleans surfaces, toxicity tests measure how harmful the disinfectant is to human cells and tissues, and residual effectiveness tests measure how long the disinfectant retains its cleaning power after being applied to a surface. Disinfectants undergo these tests in order to meet specific regulatory requirements for use in commercial settings. Types of disinfectants. There are three types of disinfectants: caustic, non-caustic, and contact disinfectants. Caustic disinfectants are those that contain corrosive chemicals that can damage tissue. Non-caustic disinfectants work by breaking down the organic material on surfaces. Contact disinfectants work by attacking the microorganisms on contact. The type of disinfectant you use is important to consider when cleaning a surface. Non-caustic disinfectants are effective against many types of bacteria and viruses, but they can't eliminate fungi or spores. Caustics can kill most bacteria and viruses, but they can also damage wood, metal, and other materials. Contact disinfectants are the most effective against bacteria and viruses, but they may not be effective against fungi or spores. Caustics are the most effective against both fungi and spores, but they may cause damage to surfaces if used improperly. When deciding which type of disinfectant to use, it is important to consider the object you are cleaning and the condition of the surface. Read the product labels carefully to find information about how long the product will remain effective and whether it is safe for use in specific environments. The procedure for disinfectant validation involves several key steps to ensure the effectiveness and safety of the disinfection process. Here is a general outline of the procedure. Selecting the disinfectant. Choose a disinfectant that is appropriate for the intended use and target microorganisms. Consider factors such as the spectrum of activity, compatibility with surfaces and materials, and any regulatory requirements. Developing a validation protocol. Create a detailed validation protocol that outlines the procedures, acceptance criteria, and testing methods. The protocol should include a clear description of the validation objectives, test organisms, test surfaces, disinfectant concentration, contact time, and any specific instructions or guidelines. Preparing the test organisms. Obtain the appropriate test organisms that represent the microorganisms of concern in your specific environment. These organisms should be characterized, pure, and of known susceptibility to the disinfectant being validated. Preparing the test surfaces. Prepare the surfaces to be tested by ensuring they are clean and free from any visible debris or organic matter. The surfaces should resemble the real-life conditions in which the disinfectant will be used. Conducting inoculation studies. Inoculate the test organisms onto the prepared surfaces using a standardized method. Ensure uniform distribution of the organisms across the surface area. Applying the disinfectant. Apply the disinfectant to the inoculated surfaces according to the recommended concentration and contact time. Follow the manufacturer's instructions and ensure that the application is consistent and reproducible. Sampling and recovery. After the contact time has elapsed, sample the treated surfaces using appropriate methods, such as swabbing or rinsing. Ensure proper recovery of the microorganisms from the surfaces to assess the effectiveness of the disinfection process. Microbiological analysis. Transfer the collected samples to a laboratory for microbiological analysis. Perform appropriate culture and identification techniques to determine the presence or absence of viable microorganisms. Data analysis and evaluation. Analyze the data obtained from the microbiological analysis, comparing the results of the treated surfaces with appropriate controls. Evaluate the effectiveness of the disinfectant in reducing microbial contamination based on predetermined acceptance criteria. Documentation and reporting. Document all the procedures, results, and observations in a comprehensive report. Include details of the disinfectant used, testing conditions, results, and any deviations or challenges encountered during the validation process. Validation review and approval. Review the validation report internally, involving relevant stakeholders, such as quality assurance and regulatory personnel. Obtain necessary approvals before implementing the validated disinfection process. Ongoing monitoring and revalidation. Implement a monitoring program to ensure the continued effectiveness of the validated disinfection process. Periodically revalidate the disinfectant as per defined intervals or in response to any changes in processes, equipment, or regulatory requirements. It is important to note that this is a general outline of the procedure, and specific details may vary depending on the industry, facility, and regulatory requirements. It is recommended to consult relevant guidelines, standards, and experts in the field for a comprehensive understanding and application of disinfectant validation procedures. Like the video, and share it with your friends and colleagues. Don't forget to subscribe to this channel and also hit the bell icon to get regular updates. Thanks for watching this video.